Please listen carefully.
Hello, Team Anaesthesia. Welcome to GasGasGas. This is the best anaesthetic science podcast for the FRCA primary exam. Our goal is to fill your brain with all this highly useful information. Now, you might be in the gym right now, commuting, or ironing your scrubs, and there's no judgment here. GasGasGas will prime your brain for the monsoon of knowledge you need to imbibe. But regardless, the revision is eventually going to end. But for now, expect facts, concepts, model answers, and the odd tangent. Now remember to check out the website, that's gasgasgas.uk. There are show notes there with all the detail, plus links to foundational reference papers and anything else useful I find for you guys. Anyway, buckle up, get ready for your mind to be bent into a new shape, and let's get on with the show.
Hello everyone, this is James at GasGasGas. Today we're bringing you yet another episode of VivaCast Action, Shyam handling the autonomic nervous system and a few other bells and whistles. I'd like to congratulate Shyam on passing his oral FRCA primary exam. Round of applause, everyone. He's passed. Now, I'm conscious that I've done a load of these VivaCast episodes, so the next episode, we're getting back into some pharmacology. We're going to dabble with ephedrine, or ephedra. That's coming in 2 weeks' time. The research has begun. Anyway, we'll let Shyam get in amongst this. Sit back and listen, and try and think about how you might approach these questions on a structural basis. Right. Physiology fun. Okay, Shyam, welcome to your Physiology Viva.
Yeah, let's go.
Break down the structure of the autonomic nervous system for me, please.
Sure. So you can break the autonomic nervous system down into sympathetic, parasympathetic, and enteric nervous system. So speaking of each, each essentially has a preganglionic section, the ganglia, and the postganglionic section. So talking about the sympathetic nervous system firstly, it's involved in your fight or flight response. So exercise, running away from a tiger is the classic example I like to use. It has thoracolumbar outflow, and it's got short preganglionic fibers, which release acetylcholine at the ganglia. And then postganglionic fibers are long, and they release noradrenaline at their effects, except for two areas. So that's at sweat glands, which are acetylcholine receptors, and the adrenal medulla, which releases an endocrine hormone, adrenaline, which is what its effects are. And then the main effects are essentially to cause tachycardia, increase the vagal tone, and essentially able to give you the energy and stuff that you need to run away. So that's the way I'd summarize the effects. I can go into those in a bit more detail, or I can explain the parasympathetic if you'd like.
Let's do the parasympathetic first, and then we'll compare and contrast their effects.
Yeah, sure. So the parasympathetic nervous system is the craniosacral outflow. So these have long preganglionic fibers and then short postganglionic fibers, and both have acetylcholine receptors, but the muscarinic receptors are what exert their effects. And again, the autonomic nervous system is involuntary, so we don't have any control over what's going on here. And again, this is in resting and digesting state. So they are to help digest food, help with that and other things as well, which they can go through. And I've talked about the neurotransmitter. I'm sure I said acetylcholine. Yes. Okay.
And whereabouts is the parasympathetic nervous system in terms of the nerves you might find that are parasympathetic?
Sure. So you have cranial outflow and sacral outflow. So your cranial outflow, so you've got your nerves 3, 7, 9, and 10. So that is oculomotor, facial, vagus, and glossopharyngeal. And then your sacral is made up from your sacral plexus, so it's usually S2, 3, 4. Those are your main ones as well. So, that's where your outflow mainly is from.
Okay, excellent. And so just comparing the effects of the sympathetic versus the parasympathetic nervous system, could you talk me through that?
Sure. So their effects are essentially mediated by G protein coupled receptors. So the sympathetic nervous system have adrenal receptors, and so alpha-1, alpha-2, beta-1, beta-2, and 3. Then your sympathetic have muscarinic receptors labeled M1 to 5. And the effects, so we'll start by system. So let's start with the respiratory system. In terms of the sympathetic nervous system, via action on beta-2 agonists, or beta-2 receptors, you have bronchodilation. So you have increased airflow to the lungs at that time, whereas the parasympathetic nervous system causes bronchoconstriction. In terms of the cardiovascular system. This is where the main effects differ. So the sympathetic nervous system causes an increase in inotropy, chronotropy, and also dromotropy as well. So heart rate, decreased diastolic filling time, and increase in systemic vascular resistance. So you have an increase in preload and afterload as well to try and bump and increase your cardiac output to your system. Whereas your actual primary innervation of the heart is actually directly innervated by the vagus nerve. So, and you have a larger resting vagal tone. So this is responsible for the heartbeat being at 60 beats per minute. And increased vagal tone will cause bradycardia, hypotension, and negative inotropy and chronotropy. In terms of your GI system, so you would have the sympathetic nervous system causes decreased gastric motility and also decreased blood flow to the GI and liver system to try and preferentially increase the blood flow to the muscles and the ones that are using to run away from said animal. And the parasympathetic nervous system has the opposite effect, so increases GI blood flow and causes increased activity and of the interstitial cells of Cajal, which causes the peristalsis that's present. Mm-hmm. Other effects, so if we talk about the eyes, for example, so the sympathetic nervous system is responsible for the dilatation that you see in eyes, and that's in essence to help you see and see where you're going when you are doing something fearful. And then the opposite occurs with the parasympathetic nervous system. And in terms of the bladder, it causes...
And I'll stop you there. That is your 5 minutes. Well done, Shyam. So you mentioned G-protein coupled receptors. What's a G-protein coupled receptor?
So a G-protein coupled receptor is a metabotropic receptor that works via a secondary messenger. It's got 7 transmembrane proteins, and essentially it's got 3 subunits, alpha, beta, and gamma, if I remember correctly. And essentially each G-protein coupled receptor is coupled to an enzymatic process or a secondary messenger which activates things downstream. So if we take the alpha-1 receptor, for example, in the sympathetic nervous system, this is coupled with GQ, which is phospholipase C. This ultimately causes the breakdown of PIP2 into IP3 and DAG, which ultimately opens calcium channels, causes more calcium to go into smooth muscles, for example. And then this was how smooth muscles contract, because they depend on calcium to undertake this. So this is how they mediate their effects on vascular tone. So the secondary messenger here would be your PIP2 and your DAG, which is what we're talking about at that time.
For extra points, PIP2 and DAG, not IP3. What are they?
Sorry, IP3. Yeah. So they're so essentially, it's what they break down. So phospholipase breaks down IP3 into PIP2 and DAG. So phosphatidyl bisphosphate and diacylglycerol is what they would be, and they are the secondary messengers.
Excellent. And what happens a panel of G-protein coupled receptors are perpetually flooded with adrenaline? What might occur?
So essentially, adrenaline acts mainly on beta-2 receptors, but they can act on alpha-1 as well. So that you can have... They need to sort of be inactivated in order to ensure that their effects aren't continual and overloaded. So there are some enzyme systems in place. For example, adrenaline's action on B2 receptors can be terminated by phosphodiesterase, which breaks down cyclic AMP, which is the secondary messenger for that system.
Yeah, okay.
And that's a way to auto-regulate itself.
Anyhow, time for a brief mention about the sponsors of GasGasGas, Teach Me Anaesthetics. These guys are behind an excellent single best answer question resource. Firstly, I took the joyful, challenging and intermittent bashing through their 1100 plus questions which they have written for the FRCA primary exam. Now I secretly loved it, and there are plenty of questions to test your knowledge. When I was studying for the FRCA primary, it did not exist, but there were other packages online. These cost a lot more, and they didn't really seem to reflect the content of the exam. Whilst doing them, I came across rehashed MRCP questions, which was not terribly inspiring and left me pretty cheesed off. Whereas these single best answer questions have been built from the ground up. They have not sniffed an MCQ in a past life. They are based on the anaesthetic science you need to know, and there are explainers with all the questions, so it builds your knowledge as you work through them. There is a multitude of ways you can study these questions. You could choose to do a random battle with 1100 plus questions, or split them into subject area. You can go back and redo the ones you've gotten wrong. It's very malleable. So if you've been thinking about which question resource you might want to get your hands on for the exam, you shouldn't really look any further than Teach Me Anaesthetics. If you reckon it's for you, all the links to Teach Me Anaesthetics from GasGasGas are affiliate links. So if you're signing up through one of those, you're helping to support GasGasGas. So if you think you're going to go for it, click a link with me. You know you want to. You'll not only be supporting your exam preparation, but you'll be supporting your 100% favourite Tell Your Nan Over Sunday Lunch podcast, which is, of course, GasGasGas.
You've smashed through that at quite a clear and effective rate. I need to now think of some questions for you here, Shyam. So it's going to be a bit of pharmacology as well. So G-protein coupled receptors, the modulation of these by anaesthetists allows us to influence heart rate, blood pressure, etc., and control homeostasis during anaesthesia. Would you be able to talk me through the different ways you can manipulate someone's heart rate and blood pressure with drugs, please?
Sure. So you can split this into sort of vasoactive vasopressors, and then so you've also got antimuscarinic medications as well. So these would all in effect increase. Well, we'll go into it a bit more first. So starting with the vasoactive, you've got vasopressors and inotropes. So your vasopressors, I would cause the sympathomimetics. So these act on the on the adrenergic receptors, and then you have synthetic and endogenous that we have. So, for example, adrenaline we can give ourselves in, for example, anaphylaxis or profound hypotension. These will directly act on the beta 1 and beta 2 and alpha 1 receptors, and these will then mediate the alpha 1 mediated increase in systemic vascular resistance. You'll have the tachycardia that you'll get with adrenaline via its beta 1 effects. Other drugs that we can use, so for example, beta specific, so isoprenaline. This we can modulate heart rate, and we can use this as an infusion in bradycardias, especially after complete heart block, as a temporizing measure before we get a pacemaker, because this will help us, because of its specific beta actions, we will be able to increase your heart rate with that. So that's an example of a synthetic drug. Ways we can try and decrease heart rate. So this could be, for example, side effects of the certain drugs. So a synthetic catecholamine or sympathomimic that we use is metoramidol. So these are mainly alpha 1 specific agonists, and they cause peripheral vasoconstriction, but this is sensed by our baroreceptors and causes a reflex bradycardia that can be seen with administration. So that's another way that heart rate, not necessarily can be manipulated, but it can be changed. And if a profound bradycardia occurs, we can start thinking about using antimuscarinic medications to try and, because this is usually mediated by increased vagal tone due to some or other reflex. So, for example, atropine, glycopyrrolate, these are all medications that stop the effects of acetylcholine.
That is your 5 minutes, sir. You were really, really slick with G-protein coupled receptors.
Thank you. Yeah.
That was just like, clearly knows what he's on about. Metabotropic, 7 transmembrane domains, secondary messenger system, all keywords that someone's going to just... of course he knows what he was on about. Right. Okay. So much so that I had to make something up.
I just wanted to clarify, what did you mean by flooding of adrenaline? Is it so hard they terminate their effects? Is that what you were after, or is that...
So, a little bit. So, it's probably worth knowing that the active, like a repeated activation of a G-protein coupled receptor downregulates the G-protein coupled receptor. Yes. I think it's beta arrestin that comes and tells the receptor to shift off of the membrane. So I think that's in part why dobutamine stops working after like 48 hours.
Yes, I was wondering why dobutamine did show tachyphylaxis, but I'm guessing that's how it does that.
I think so, yeah. Yeah, it just gets knackered. And that's why other routes to making blood pressure go up again may or may not be necessary. But it means that dobutamine itself means that it self-titrates off. Because if your patient's still got a heart that isn't pumping due to like a septic cardiomyopathy after 2 days, then like it's probably quite bad news, isn't it?
Yeah.
And that is Shyam, again, demonstrating that he has really put the work in. And you have to. It's a proper hard exam. And you want it to be a proper hard exam because we want robust examination of anaesthetists who could accidentally kill someone very quickly. Most other specialties can't accidentally kill people that quickly unless you give a drug incorrectly. If we goof it because we're not organised and we're not on it, the poor results are rapidly fed back with a blue patient, etc. Train hard, fight easy. We've got to do the studying. So I want to say, keep studying for this exam, keep going. It is a marathon, it's not a sprint. A little bit every day is much better than trying to smash it all at the end, unless you're one of those people who can somehow do that. I don't think they exist. I think they just secretly study a lot but don't say they do. We'll see you next time.
Ahoy, Team Anaesthesia! You've survived yet another episode of Gas, Gas, Gas. Now, if you found it useful or harrowingly awful, please like and subscribe, drop us a star or 12, and follow with whichever podcast platform you find yourself using. Please leave a comment or ping off an email if you think I need to square something away. Now, there are a bunch of ways to support the costs of Gas, Gas, Gas. From buying me a coffee, to venturing forth via an affiliate link, to the horde of joyful SBA questions from Teach Me Anaesthetics. Those links are on the website and in the show notes. Speaking of website, definitely check out GasGasGas.uk for the show notes, diagrams, details, the references. Now, we all know, guys, that this is a bucket of content to consume, and it is like drinking from a fire hose. So I want to finish by saying, take it day by day, don't overcook yourself, don't freak out, and keep studying.